Hello, this is Bioemission Technology Solutions, and today we'll be discussing how our new I-Series, the only completely portable, affordable, small animal full-body imaging system, can help facilitate and improve the quality of studies of numerous groups having access to radioisotopes. We will talk about the benefits of using the eyes by groups conducting biodistribution studies, users that have access to clinical cameras, limited access to high-resolution multimodal systems, but also those that own such a high-performance system. So let's start. Our eyes products are dedicated systems for in vivo molecular imaging of radiopharmaceuticals, radio-labeled biomolecules, and nanoparticles. Beta Eye is the only coincidence PET imaging system which is actually a planar PET. On the right-hand side, you can see some of the images of the standard clinical tracer FDG taken with Beta Eye in a normal mouse. Our second system is the Gamma Eye, the only scintigraphic system in the market, a planar SPECT system. Here you can also see some typical images of the clinical bone tracer Technetium 99NDP acquired with Gamma Eye. Both systems provide fast screening at a very competitive cost, at least 5 times lower than a multimodal system, thus making it easily accessible to a wide number of researchers. They are benchtop and fully portable, very easy to operate by non-experts, and fully designed based on end-user needs. So how can we maximize the quality and effectiveness of studies for research groups? Being researchers and academics ourselves, this has been one of the main concerns for at least the last 10 years. Biodistributions are a standard part of a preclinical study. Both groups that own an imaging system and those that do not own one use biodistribution studies to identify the accumulation of a tracer at specific organs. The standard practice is to obtain data like the one shown here, which are graphs that include the accumulation of radioactivity measured at different organs and is proportional to the tracer's concentration. To obtain such a curve, one has to sacrifice several animals at different time points, for example as shown here, at 1, 2, 4, 6, 8, up to 24 hours. Do this for at least 3 animals or even more in order to have accurate statistics, and then plot the average number of counts for different organs. However, molecular imaging with a planar system allows you to obtain a series of images from the same animal, draw regions of interest on the organs of interest, and extract semi-quantitative curves, which are similar to the graph on the left, for various time points by just using one animal for all those points. So in order to perform a long study, in the end, you would need a small portion of animals compared to the biodistribution study. Furthermore, each animal is used for all its time points, so it can serve as a control of itself, thus increasing the accuracy of the results. Although advanced users question the role of planar imaging, one of the most widespread procedure for easily studying a new tracer in vivo is the use of optical imaging. So this is a well-accepted technology for imaging a biomolecule over time. However, it has certain limitations. Optical imaging has low spatial resolution and cannot provide quantitative information. In many cases, the tracers for optical imaging need different chemistry compared to the tracers that are used for radioisotopes. Fluorescent dyes may be large molecules that may alter the biodistribution of new biomolecules and in some cases are toxic. Also, many groups that use radioisotopes do not have access to optical imaging. Costly, dedicated equipment is needed, and also one needs the experience to use fluorescent dyes. Furthermore, groups that use radioisotope cannot immediately translate their results to imaging if they have to use optical imaging first and then radio tracers. Finally, optical imaging has low penetration depth, so this technology cannot be used in larger animals, starting from rats and going to rabbits or even larger animals. So if you develop a tracer and want to study it on mice with optical imaging, you definitely need another method to go to larger animals and proceed towards clinical research. Finally, it is very important to emphasize that the cost of an optical imaging system is higher than the cost of gamma and beta I combined. Thus, groups working with radioisotopes do not need to pass through optical imaging for obtaining the first in vivo imaging data. Most of the published studies where small animals are used are related to oncology. In these cases, tumors are usually located on a mouse's foot and do not overlap with other organs. The first question that researchers have to answer is if there is targeting, and then understand how optimum targeting can be achieved. In this PET tomographic image, a tumor placed in the upper left foot of the mouse can be clearly seen. However, there are not major advantages in using PET imaging, as even with optical imaging, which is not quantitative as already mentioned, this information can be quickly obtained and the accumulation on the tumor can be clearly seen. What we suggest is to use planar imaging to both extract this information but also get quantitative results, since by drawing a region of interest around the tumor, one can quantify the accumulation as a portion of the radioactivity over the entire mouse. Furthermore, you can perform dynamic imaging, and as can be seen here, it is possible to observe how the tracer distributes over time in the animal, something not possible tomographically, where very fast tomographic images cannot be obtained. Quantification is a strong advantage compared to optical imaging. The first step is to verify that tumor targeting has been accomplished. However, tomographic imaging is not needed to do so. Here, 4 examples of a tumor placed on the upper left foot can be seen. In the first image, no accumulation on the tumor can be observed. On the second image, a low accumulation on the tumor can be seen. On the next image, we can see high accumulation on the tumor, but also high background on the mouse. And on the fourth image, we can see a very specific accumulation on the tumor, no background of the mouse's body, only some background on the intestines, and of course accumulation on the bladder, meaning that in the fourth case, we have achieved the best targeting. So this information is very clear, can guide the researcher for the next steps, and is obtained just by using planar imaging. Most people that perform tomographic mouse imaging usually do not study all the conditions for animal preparation prior to ex vivo biodistribution and imaging. However, there are several parameters that can affect the biodistribution, such as food and water consumption or body temperature. Images are usually obtained by following only one protocol without fully examining all these conditions, since it is not practical to assign a large number of animals under different conditions and do that on tomographic mode, since tomographic time is always expensive. Published studies demonstrate that many biomolecules have completely different behavior depending on animal preparation, which should be part of the studied protocols. So we believe that this very important step, usually missing from most studies, can be easily achieved if the user has a small planar tool like gamma and beta I. So if we were to summarize all the questions that someone should answer before concluding whether a tracer works well or not, we could have a list like the one here. Do biomolecules reach the target? When we inject a substance, does it go where it is planned to go? Are biomolecules accumulated in other organs or tissues? Is it likely that we target organs that we shouldn't target? For how long does a tracer remain on a target? If it is a diagnostic or therapeutic tracer, this information is very important. For how long do biomolecules stay in the blood circulation? Many times targeting is low, so we need to ensure that the tracer stays for a long time in the blood circulation. Are the tracers stable post-injection? Most of the studies are done firstly in vitro, but when we inject a substance in a living organism, several parameters may affect the tracer. It is vital to have an indication that the tracer is stable post-injection. What happens during the first minutes post-injection? It is very important to understand and obtain images, if possible, from the first blood pass to understand what happens when the tracer starts circulating in the body. Is it trapped in any organ? Is it eliminated very fast? What is the best injected concentration to optimize image quality and reduce dosimetry and toxicity effects? How shall we prepare the animals before imaging to optimize tracers' biodistribution? When is the highest accumulation in the target to define optimal imaging points? That is, if we plan to do tomographic imaging. When is the best time for biodistribution points, and how can we optimize the necessary number of animals? Also, which is the best injection route, especially for new products and new applications? So there are numerous questions that certainly most researchers do not answer, although they should, and the main reason is the lack of a tool that can easily provide images and allows them to test different parameters before starting to use a large number of animals. And this is exactly what the eyes can offer. So, is tomographic the first step? No, we certainly think not. So what is the current situation in terms of usage applications as gathered from the latest publications? Following an initial market analysis, we have realized that 75% of groups that publish studies using small animals and SPECT radioisotopes do not have access to high-resolution images. It is surprising to find that only 26% of the published studies from more than 40 journals have images from multimodal tomographic systems. 11% of the groups just perform stability studies in cells. 34% of the groups work with biodistributions, and although they could obtain images, they do not have a tool to do so. Also, 29% of the groups are using clinical cameras, which have poor resolution but can give an indication of the tracer kinetics. Even the groups that use multimodal systems do not necessarily own one, but usually rent it for limited time and number of animals. So there is a clear need in the market and in the research community for tools that can provide good images, but in an affordable way that can become a standard daily tool in every lab. After this introduction, and now that we have hopefully triggered your interest, let's go through some more specific examples that show the usefulness of both gamma I and beta I for a group performing biodistributions, so that this group can have in vivo images as extra data, but also significantly improve the way they perform biodistributions. The obvious advantage is the extension to in vivo images. The procedures of performing biodistributions are very standard and established. The animal needs to be sacrificed, the organs are extracted, and then, by using a gamma counter, the radioactivity in each organ is measured, and so we have a clear indication of the radioactivity accumulation per organ inside the animal. This procedure is straightforward, but it has some strong disadvantages. For every time point, several animals need to be sacrificed. At least 3 to 5 animals need to be used for each time point in order to have acceptable statistics, since each animal is used only once and the biodistribution in each animal may vary. The cost to purchase, host, and use all these animals for one study is far from negligible. If in one biodistribution study we need 100 animals and in another one, only 10. The cost is increased by a factor of 10. The procedure of doing a biodistribution takes time, as well as the procedure to process and analyze data. Usually, all these time points are not taken on the same day and not from the same batch, so this procedure is even more complicated than it seems. And let us not forget that time costs in a lab, because it is time for personnel, time that someone could use to do something different. Thus, this procedure does not facilitate the test of many ideas in animals, and we have to carefully select which animals will go through biodistribution. Last but not least, we have to be in compliance with bioethics, which have become stricter and require the use of fewer animals with good justification. It is very important to respect the 3 R's principle. On the other hand, in vivo planar imaging, we can use only one animal to obtain a full data set for all time points, draw regions of interest around the organs or tissues of interest, and extract time activity curves for one animal. So, if we have a group of 5 animals, we can repeat the same study with very good statistics and obtain an accurate time activity curve over a long period by using a small number of animals having the same animal for all time points. This significantly improves statistics and, of course, accuracy. Easily test different administration routes and check biodistribution. The administration route is a dominant parameter when studying new biomolecules. While most of the studies used to be intravenous, different administration routes are now examined. Especially for new biomolecules, including nanoparticles, different administration routes should be tested. It is well known that the biodistribution is significantly affected by the administration route because the tracers go through a different route and pass through different organs. Planar imaging can easily visualize the differences in a biodistribution over a long time, and so by using a small number of animals, the researcher can test different administration routes and define the final imaging protocol. So the eyes can guide you for optimizing the administration route for your ex vivo and in vivo protocols by using a minimum number of animals. Optimize animal preparation conditions before starting the full protocol. Even for standard radiopharmaceuticals, several parameters can affect biodistribution. The most important parameters can be fasting, heating, and anesthesia. Of course, it is not possible to create a group of 30 animals and use biodistribution to study all of them. And this is the reason why most groups do not study how different preparation parameters affect the biodistribution fate of their tracers. It is likely that wrong parameters are used that affect the results and in many cases cause misleading conclusions about the success or not of a new tracer. Planar imaging can easily visualize the effect of different preparation conditions by using a small number of animals and testing a few parameters before going to the large animal dataset. Again here, the eyes can guide you for optimizing your ex vivo and in vivo protocols. Using imaging in a few mice to select the proper time points for biodistributions. No one doubts that biodistribution gives significant and accurate information and will remain a key step for testing a tracer. The question is when to obtain the biodistribution points. So when studying new biomolecules like nanoparticles, usually the behavior is initially unknown and we may have evidence of what will happen or we may have no clue. Planar imaging and dynamic studies easily allow the researcher to obtain a first impression of what happens. By using a small number of animals, we can obtain this information, and this allows efficient animal use as well as deciding on further evaluation or not. This significantly reduces cost and time required to understand how a new tracer behaves. In addition, we use a reduced number of animals, which means that either we save resources or we can use a small number of animals for studying different parameters and for testing more ideas. In this way, we are clearly in compliance with the 3 R's principle, so the eyes can guide you for selecting your ex vivo biodistribution time points with a minimum number of animals. Check all mice that participate in a biodistribution study and exclude bad injections or animals with strange behavior. When performing biodistribution studies, we usually inject animals with our tracer, then sacrifice and start counting the dose in each organ. By using the eyes, it is possible to image all mice that participate in a biodistribution study before sacrificing them or right after. So we can identify bad injections and we can exclude these animals in order to improve statistics. In addition, we can identify aggregations or other unexpected accumulations which are visible in imaging, but we would have no evidence that they occurred if we only performed biodistributions on selected organs or tissues. When we exclude animals with obvious errors from biodistributions, we save cost and time of analyzing animals that give us no information. We increase the accuracy of the statistics. We improve the standard deviation. And so in the end, we need far fewer animals to obtain good statistics. If we have a certain number of animals available for an experiment, we can test more parameters with the same resources, and again, we are in compliance with the 3 R's principle. So, the eyes can be used as a quality assurance tool that can change the standard way of performing biodistributions because it can image all the animals that are participating in the biodistribution before proceeding to the ex vivo study. Identify unexpected areas of accumulation. When we perform biodistribution studies, we are usually interested in certain organs. However, if we have the ability to obtain whole body image of a mouse, as we can see in this example on the right-hand side, it is likely to identify in some cases unexpected results too. For example, in this mouse, we notice an accumulation on the left bottom foot of the animal, which was not anticipated. We wouldn't have evidence that this accumulation existed if we did not have the opportunity to obtain dynamic images. We have the ability to visualize an entire mouse in a single view as a function of time, identify accumulations at different regions, and suggest other tissues or organs that should be included in the biodistribution study. So the eyes can identify unexpected areas of interest when studying a new biomolecule or procedure. Optimize injected dose in terms of concentration. The injected concentration definitely affects biodistribution, but is a parameter that most groups do not study in vivo, although it is critical in translational research. However, it is vital to define optimal injection concentration when testing a new biomolecule with unknown behavior. Low concentrations may not allow sufficient accumulation at the target, so may not give us a sufficient imaging signal. On the other hand, high concentrations may saturate receptors and increase background or signal in other organs. So by using planar imaging, we can easily visualize the effect of different injected concentrations. As we can see here in these 3 examples, on the top we have injected the same tracer and tested 3 different concentrations of 0.05, 0.1, and 1 milligram per milliliter. On the left image, we have a minimum concentration in the body. On the right image, we can see the contour of the mouse, which means that the higher concentration we injected have created an extra background on the animal. In this way, we can understand what the proper injected dose is and perform the optimal biodistribution studies. So the eyes can be a tool to guide you to optimize your injected concentrations and optimize your ex vivo and in vivo protocols. Optimize injected dose in terms of radioactivity by doing tests on a few mice. Besides optimizing injected concentration, a parameter that we need to study is the optimization of the injected dose. Differences in injected radioactivity can affect the biodistribution and imaging performance for diagnostic tracers. For example, low activities may not allow target visualization. High activities may saturate the imaging system or may increase the background. So we have to decide on the best radioactivity to inject. In many cases, low injected activities cannot be avoided due to a low radiolabeling yield, so we have to be sure that the image information we acquire is satisfactory. In repeated studies on the same animals, we may decide to inject low activities to ensure that there aren't any dosimetric issues. Planar imaging can be used as a simple tool to provide information on the images that we get by injecting different radioactivities. So the eyes can guide you to optimize your injected radioactivity in ex vivo and in vivo protocols. Planar imaging is the only way to check the first blood pass. When we study a new tracer, it is very critical to know its behavior immediately post-injection. This is something we cannot do with tomographic systems, and clinically it is done in planar mode. Even with the advanced multimodal systems, it is not possible to obtain the first blood pass image because the field of view of the animal does not allow a whole body image, and the time that we need to perform tomography is more than a few minutes, resulting in a loss of the first blood pass. However, the eyes, which have a field of view comparable to or bigger than a mouse, can perform the following steps. Place the animal on the system, start the acquisition, and then inject the tracer. So we can take fast images down to 10 seconds per frame, as you can see on the right-hand side. When we do this for a few seconds or minutes, we can sum all the images, draw regions of interest on the sum of these images, apply these regions of interest on all the frames, and draw time activity curves from these ROIs to understand how the tracer behaves. This is very important because this information cannot be derived from biodistributions, as we cannot take biodistribution points every few seconds. Also, as already mentioned, this cannot be done with tomographic imaging, so the eyes can provide whole body data from the first second post-injection. Fast data during the first minutes post-injection requires many animals for ex vivo biodistributions. It is also very important to obtain information during the first minutes post-injection. With a planar system, it is possible to place the animal on the camera, start the acquisition, inject the tracer, and then select a protocol which will provide fast images for the first frames and subsequently increase the duration of the imaging frames. For example, this protocol can provide 10 seconds images for the first minute, 30-second images for the next 3 minutes, and then 60-second images for the rest of the study. Again, we can draw the regions of interest on the sum of all these images where all the organs are visible. We can apply them to the first frames. We can draw time activity curves to understand the tracer's behavior, and in this way, we can use very few animals to obtain many initial time points, with each animal as a control for itself. This is something we cannot easily perform with biodistributions because it is not easy to sacrifice animals with a difference of a few seconds, and of course it is not possible to do this with tomographic imaging. So it is important to understand that the eyes are the only tools that can non-invasively quantify and visualize the biodistribution of a tracer during the first minutes post-injection. Design pharmacokinetic models. Pharmacokinetic studies are a critical step when testing a new tracer. They are usually performed using tomographic data. However, there are many cases when such a model can be derived from planar imaging. The eyes allow us to obtain a series of dynamic images. We can then extract time activity curves for the major organs, tumors, or other tissues. It is important to use organs that do not overlap in the planar images. These data are semi-quantitative, and for non-overlapping organs, in practice, they are quantitative. We can correlate them with biodistribution data, which are more accurate, and we can validate a kinetic model. We can have biodistribution points at certain frames, compare them with the time activity curves from the images, and in a way correct the time activity curves for all time points. So even with planar imaging, we can have a good indication of tracer kinetics at different regions, and if we have access to a tomographic system, we can also correlate these data with the tomographic ones. So the eyes can be an efficient tool for assisting kinetic modeling studies of new biomolecules. Image all organs at once ex vivo. A biodistribution study is based on selecting different organs and using a gamma counter to measure the radioactivity they hold. Similar studies have been performed with optical imaging, although they are not quantitative. It is interesting to think that such organs extracted for biodistribution can be placed either on a gamma eye or on a beta eye and be measured, so we can immediately obtain the dose per organ without the need of a gamma counter. If we weight the organs, we can also extract the dose per gram for these organs. So the eyes can be an alternative and fast tool for performing ex vivo biodistribution studies or imaging organs post-mortem. To conclude, the eyes are definitely an easy way to obtain images and shift to in vivo. Researchers can reduce the number of animals needed, time, and cost. More scenarios can be tested with the same resources while being in full compliance with the 3 R's principle. Through our market research, we found that almost 30% of groups publishing research in the testing of new products with radioisotopes use clinical cameras for imaging small animals. So what can the eyes offer to those working with clinical cameras? Let us see. Replace the blurred images of clinical cameras with images of good spatial resolution and quantified information. The problem here is that these clinical cameras produce blurred images with poor spatial resolution and practically no quantified information. With the eyes, researchers can obtain high-resolution images with all time points using just a few animals. Feasibility to image all mice, since frequent access to hospitals is not easy. Apart from producing low-resolution images with no quantitative data, use of clinical cameras cannot be a standard practice for researchers due to practical constraints. It is often difficult to get formal permission for performing mice experiments in clinical environment. Publishing these results is becoming less straightforward due to ethical restrictions. The eyes can solve all these constraints. Bringing the eyes close to the clinical camera for direct comparison, that is, if CT is also available. Having access to several clinical radioisotopes allows, in principle, physicians and practitioners in every hospital to take advantage in preclinical research. The bottleneck today is the limited access and inefficient performance of existing clinical cameras. The eyes are rather small. Their competitive cost allows them to be purchased as a complementary device and then be easily installed close to existing clinical cameras. Besides their standalone use, it is possible to take advantage of the CT component of a clinical SPECT CT or PET CT and combine this anatomical information with the functional information that the eyes provide, thus obtaining multimodal images. It is also possible to perform a tomographic scan in the clinical system at the optimal time point, which is defined by the eyes during a dynamic study. The eyes are the only systems in the market that can easily be installed in a clinical room, be combined with clinical cameras, and for the first time allow clinicians in every hospital to perform preclinical research. To sum up how groups performing imaging with clinical cameras can benefit from the eyes, the ability to replace the blurred images with high-resolution quantitative ones is definitely important and minimizes complexity. But also the eyes allow for the first time clinical researchers to easily perform basic research in a clinical environment. Even with limited or full access to a multimodal imaging system, no doubt there are numerous benefits and applications for using the eyes complementary. So all the things discussed before, plus the ability to use efficiently the multimodal imaging system and increase throughput, that is the work rate. Even if you have access to a multimodal imaging system, you definitely perform biodistribution studies as well. In the previous slides, several examples showing how the eyes can facilitate and improve the accuracy of biodistribution studies have been demonstrated. You will see how the eyes will let you test more ideas regarding preparation conditions, optimization conditions, and finally decide which animals should be imaged and when. Even if you have access to a very advanced multimodal imaging system, it is not possible to test all your products, all your protocols, all your ideas on this system. The main reason for this is that both scanning time costs and usually a system is reserved for a specific duration and number of animals. The standard practice shows that the groups that have access to a large multimodal systems do not image different preparation conditions. They do not test different administration routes. They do not study alternative concentrations. They cannot image the first minutes post-injection. They cannot obtain whole body images easily, as in most cases, the components inside the multimodal systems should be switched. However, the eyes are simple tools that are designed specifically to allow you to test all your ideas in your lab and decide when and how to scan on a multimodal system, and thus fully exploit its use for the benefits of your research. So some of the advantages are the fact that you can assess on imaging level different preparation conditions using a few animals, that you can image alternative administration routes easily, that you can image the effect of different concentrations, that you can obtain images immediately post-injection in an easy way, that you can have very short frames to help you understand the tracer kinetics. The system is designed in a way that it provides whole body images. So before using a multimodal system, you can decide in your lab on the best timing of their use, having the optimal animal preparation and the ideal injected concentration. In this way, it is possible to use a multimodal system only for your optimized conditions and best products, thus increasing its throughput. Use the multimodal system for best products and optimized conditions. On the left image, we can see different tomographic images during a PET scan of a mouse, which is either fasted or not fasted, and is injected or not injected with insulin. It is clear that certain parameters significantly affect the biodistribution, but it is expensive to study all these parameters in tomographic mode. However, as we can see from these images, this is very important because we observe clear differences in the biodistribution of the mouse when the same parameter is studied on animals with different preparation. In practice, chances are that you would not decide to buy imaging time in a multimodal system for testing all those scenarios. On the right-hand side, we can see planar images of the standard TC99M MPD for bone imaging and the effect of different parameters with gamma I. The effect of most parameters is clear in planar mode. In addition, it is fast and easy to test different protocols on a few mice. It is easy to study the effects as a function of time too. So there is no need to buy imaging time for performing all these tests. You can decide on planar mode what the best parameters are and then perform 3D scans in an optimal and efficient way. Image the first seconds, minutes post-injection, which is not possible in tomography. On the left image, we can see a high-resolution tomographic SPECT image of a mouse at 1, 2, 4, 24, 96 hours post-injection. The disadvantage of tomographic imaging is that it needs at least a few minutes to be performed, especially for SPECT. So it is not possible to track fast phenomena, especially at the first minutes post-injection. It is not easy to perform the 3D scan on the same animal multiple times, especially if you do not own a tomographic system and you have to schedule your access to one. It is also not easy to perform 3D scans in many animals because of the time cost. It is not practical to occupy a multimodal system just to obtain these time points. On the other hand, by doing in vivo planar imaging with either gamma I or beta I, it is possible to obtain all time points right from the first second post-injection, since the system allows even 10-second frames to be obtained. It is easy to perform a dynamic study for the first 60 minutes post-injection and then perform static imaging at 1, 2, 4 hours as the system is right in your lab. It is feasible to study multiple animals in parallel by properly deciding on the time points of the injections, the time that the first dynamic study will be completed, and the time difference between the injections of the different animals. There is no need to buy imaging time for this procedure. You can complete the study and then decide if an additional 3D scan is required and what the optimal time point for it is. Define the best time points for advanced tomographic scans. It is not practical to perform 3D scans at various time points, so it is critical to have an easy way to define the best time point to do a tomographic scan. As you can see in the images at the upper right, in all cases there is a tumor at the left bottom foot of the mouse. In the fourth image, accumulation can be seen only in the kidneys and tumor, while in the other images we have also significant accumulation at different organs. So we can decide that the last image is the one that better demonstrates tumor targeting. By doing planar imaging with gamma I, it is possible to understand the kinetics of the tracer and then define the best time points for performing a tomographic study, thus minimizing cost and time related to multiple unnecessary tomographic scans. We can then schedule access to a multimodal imaging system at the optimum time points. In this way, the eyes can be your daily tool to define the optimal time points before proceeding to advanced tomographic scans. In this example, you can see a tumor which becomes visible at 20 minutes post-injection, which means that tomographic imaging should be performed at least at 20 minutes post-injection and not earlier. Verify that an injection is successful before a tomographic scan. Even when we perform imaging scans on advanced imaging systems, there is a need of a tool to confirm that the injection is successful, that the tracer has entered the animal, and that the tomographic imaging is worth doing when the tracer concentrates on the target. As you can see in the right image, we have two examples of bad injections, as imaged with gamma I immediately after the injection. In the first image, the tracer has not entered the animal at all, and in the second, the tracer is inside the animal, but a high accumulation can be seen in the tail vein. Thus, both animals will not provide accurate tomographic data. Doing a tomographic scan immediately post-injection is not a good practice, since it takes time and it occupies the system instead of allowing another study to take place. So it is extremely useful to have a tool that validates the quality of injections in real time, easily, and confirm if an animal is suitable for tomographic scan or if we need to select another one instead. Having the eyes as a screening tool beside a multimodal system can definitely improve its throughput. Even with an advanced tomographic system, it is not possible to image the first seconds or minutes post-injection. Again, it is a fact that in planar mode, we can start imaging with injection and that we can take fast images immediately post-injection. This is something that cannot be done with tomographic imaging. The eyes are the only tools that can non-invasively quantify and visualize the biodistribution of a tracer at the first minutes post-injection. Define the best time points for advanced tomographic imaging. It is very important to decide when to perform tomographic imaging, especially for new tracers. It is critical to know in advance its biodistribution, the maximum accumulation on the target, and then select the optimum time points for 3D imaging. Tomographic images will provide quantitative data that can be used for scientific, publication, and promotion reasons. However, it is not easy to find the optimum time points by doing tomographic imaging, especially for tracers with unknown kinetics. The eyes allow us to study a tracer easily in planar mode and decide when to image the mouse at a multimodal system for getting the 3D images. On the top image, we can see that the mouse is continuously scanned on planar mode. The time of the maximum concentration on the tumor can be defined at around 90 minutes post-injection, so a SPECT scan can be arranged to be done exactly at that time. In the bottom two images, we can see a high-resolution SPECT. In the first image, we have an accumulation on the tumor, but also in other organs. On the second image, we have accumulation only in the kidneys and the tumor, which is definitely the one to use when publishing. We should keep in mind that a 3D scan usually requires several minutes to be performed. In practice, the animal is usually sacrificed during SPECT to stop kinetics. This is a very significant parameter because the deceased animal can no longer be further utilized in the study. Furthermore, the SPECT time points are pre-selected before bringing the animal to the multimodal imaging facility, and no changes are possible. By having a tool that provides an indication of the best time points in every lab, researchers can significantly improve their daily work. Select the mouse with the best biodistribution for proceeding to the tomographic imaging. When we test a product, it is likely that we have different behaviors on different animals, and that the biodistribution on the same tracer is not exactly the same on all animals. So, assuming that we have injected 3 animals, it is important to decide which one we will scan tomographically in order to obtain the best quantitative data for scientific, publication, or promotion reasons. On the left part of the image, we can see an example of 3 mice injected with the same tracer, but with different biodistributions as imaged by gamma eye. It is not practical to scan all of them and then decide which one to keep. If we do it, we will have to pay for 3 different animals and assign imaging time to 3 different experiments. The eyes allow imaging of all candidate animals in a very simple and efficient way, and through planar imaging, select the optimum that will proceed to the tomographic scan. For example, from the 3 mice on the right, it is easy to perform a planar study on all of them and decide to do tomography only on the 3rd one. No need to perform a tomographic study in order to generate a whole body image. Very often, a planar image in the end is necessary to better demonstrate the biodistribution of a tracer. The current tomographic systems have a field of view smaller than a mouse. They allow a sequential or spiral scanning of the mouse, and in each case, a part of the mouse is inside the field of view. Then it is possible to do tomographic reconstruction, add all the slices, and obtain a planar image, which is the sum of all slices. This image needs several minutes to be derived, so we cannot use this method to perform fast dynamic studies or image the first minutes post-injection. In addition, it is not efficient to occupy a tomographic system just to obtain a planar image. The eyes are the only tool in the market that can easily and non-invasively provide whole-body dynamic images. No need to adjust the system for whole-body imaging. Some multimodal systems offer the option to change the collimators to whole-body imaging. However, it is not easy to implement this as a standard practice in the daily workflow, especially if the users of the system are not experienced. Very often, in order to have whole-body imaging, we minify the object, thus we significantly decrease the resolution and sensitivity. In addition, the user cannot design specific protocols for fast dynamic imaging. Definitely using an expensive multimodal imaging system for obtaining planar images is not efficient. Again, the eyes are the only tools that can easily and non-invasively provide whole-body dynamic images because they are especially designed to do so. Perform more than one scan and correlate limited tomographic time points with frequent planar time points. If you have access to a whole-body tomographic system, there are many interesting things that you can do together with the eyes, which will change your workflow. For example, you can image your mouse with the gamma eye, as you can see in the image on the top, and obtain all time points fast during the first hour. Then, you can do short scans in planar mode with gamma eye, but also tomographic scans with your high-resolution SPECT system. From the SPECT system, you can have the 3D information, quantify with greater accuracy the tracer distribution at the different organs, and correlate this information with semi-quantitative information that is derived from the gamma eye in planar mode. In this way, you can correlate the semi-quantitative kinetic curves of gamma eye with the quantitative curves of your SPECT system, and in the end, obtain time activity curves for a large series of time points. The eyes introduce a new way of exploring tomographic systems for performing dynamic studies in 3D, and there are definitely many applications to explore. To conclude, the eyes are simple to use, low cost, save time by avoiding unnecessary scans, plus you can test more products with the same resources. They will definitely change your workflow. This is it for now. We hope you found this interesting. For more information on our eyes, send us an email or give us a call. We will be happy to hear from you. Thank you.